We have a not in person, but a Zoom presentation today from Ireland. And with that, I would like to ask Dominic to introduce our speaker, please.
Thank you, Chris. Good morning, everyone. My name is Dominik Lewandowski, and it's my honor to introduce our esteemed speaker today, Dr. Imre Lengyel. He will enlighten us with his talk, The Road to Uncovering the Real Meaning of Drusen. Dr. Lengyel is a professor of translational ophthalmology at Queen's University Belfast and honorary senior fellow at University College London in the United Kingdom. Dr. Lengyel earned his PhD in medicinal chemistry in 1992 from Albert Szent Györgyi Medical School in Hungary. And then he pursued a postdoc at the University of Newcastle, Australia. After finishing his postdoc, he returned to Hungary in 1998 to establish his laboratory at the Biological Research Center of the Hungarian Academy of Sciences to study learning and memory processes in Alzheimer's and other neurodegenerative diseases. In 2006, he was invited to set up a research program at University College London to find correlations between brain and retinal degenerations. Dr. Lengyel's pioneering research on AMD, particularly the role of metal ions like calcium and zinc in drusen deposit formation and disease progression, has earned him widespread recognition as an authority in the field. His work first identified hydroxyapatite, which is a specific calcium phosphate mineral, as a key component of drusen, advancing our understanding of AMD pathogenesis. Over the past decades, Dr. Lengyel has built an outstanding research portfolio, making significant contributions to ophthalmology. His influential work has been published in top journals such as Science Translational Medicine, PNAS, Progress in Retinal and Eye Research, and Ophthalmology. He also received prestigious awards, including Bill Brown Charitable Trust Senior Research Fellowship and Charles Goddard Research Award from the National Heart Foundation of Australia. With that, let's warmly welcome Dr. Imre Lengyel to our seminar series.
Thank you very much for this very kind introduction, and especially Christoph for inviting me and Gruzina to organize this seminar. It's a very interesting journey, what you heard there. How do you get from the brain to the eye? But it's not what I'm going to talk about it, but very big proportion of the work that I will present today is going to relate to some way or the other to what we are doing in looking at what happens in the eye in diseases like Alzheimer's disease, multiple sclerosis, and Down syndrome. So this story is more about what drusen is, what we learned about it, and I gave a very big title, The Meaning of Drusen, or Meaning of Life, but it's hopefully not going to be completely hanging in the air at the end. There are some answers which we could gather so far, but there is a lot more to do about drusen and how does that develop. But I'd like to start the journey at the very beginning when I moved to London, and I was incredibly lucky to work with Alan Bird and Tunde Petu to start to understand what drusen is in a clinical setting, because that is the kind of correlation with these extracellular deposits that drove me from the brain to the eye and then eventually back to the brain in this translational journey. So what you see in this picture is something that you are all very familiar with, an OCT image where you can see where the drusen deposition is. Of course, at the time when we started, OCT wasn't as good to resolve these kind of details. So we rather relied on, especially a biochemist, cell biologist like myself, on what's happening on cross sections. And what you see in the bottom page, hopefully the cursor is visible, is this big blob between the choroidal vasculature and the retina. And even though the blob itself is in the way, the otherwise the retina is relatively healthy. There is retinal pigment epithelium, photoreceptors, and all the regions what you expect to see there. Now, the question is that, which is still reverberates, but I think is becoming more and more significant at that time, was that is there any role for these deposits? Is drusen, we know that is associated with AMD, but is it actually important, or is just simply an association? And hopefully I will show you a few images what might be happening when you have drusen deposition. So the journey started to understand what the eye is. Remember, I came from the brain, so I didn't know what the eye looks like. So in my journey, started to flat mount eyes from different ages and try to understand what is it that we can see if we remove the retinal pigment epithelium. What you see here is the first paper I published in the ophthalmology domain, where we just flat mounted some tissues and looked at what we can see. Now, what you see here is, although the neurosensory retina is removed, the RPE is still largely present. And the first observation which I made, and I'm sure this is not that surprising to you, but it was to me, although this is with age, the number of drusen increases, but it's not a rule of thumb. So you can see people at very old age with few drusen only. But this is to remind you is when the retinal pigment epithelium is still in place. So we took pairs of eyes and started to look at what happens on the top row when the RP is still present, and what happens if we brush away the RP and just look what's underneath. And what was very, very clear is that when the RP is present, we often don't see these drusen. Now, these are subclinical drusen very often, so these are not the big soft drusens that we often associate with disease. These are tiny deposits, but the importance is that they are very widely present, even if we clinically don't see them. Another interesting observation was that even if the macula, which is labeled with a star here, or it would be in the middle of this non-drusen deposited areas, the peripheral retina has a vast amount of drusen deposition, and that eventually led to many publications on looking at peripheral changes in diseases like Alzheimer's disease. And we have now fairly good understanding that both drusen deposition, vascular changes, and so forth seems to be associated in the peripheral retina with disease more than when you just concentrate on the macula. But what does this mean? Why drusen is deposited? This is the same the same paper where we looked at this drusen depositions on these flat mounts with fluorescent microscopy, just simply looking at autofluorescence of the tissue. And as you can see that every places where drusen is present, the RP adherence to this deposit are pretty poor. Surrounding it, there is good adhesion to the Bruch's membrane, but as soon as you have drusen deposition, then the RP starts suffering. At least that was our conclusion. So it might be quite important to look at this this phenomena. How does this develop? How does it grow? What is the base behind drusen formation? Now, of course, we can brush over the RP and we can visualize the deposition, both the tiny and bigger ones. And if I draw your attention to figure J, what we found is that every time a small drusen is always on the pillar between the two choriocapillaris. And that, of course, means that there is or likely to be a clearance problem, and that would explain why drusen starts on these pillars, and I think it still stands quite strong. And the lower pictures are just different parts of the flat mount in the macula, in the mid to far periphery, and every time the drusen deposition seems to be aligning with the choriocapillary pillars. Now, it was also interesting when you look at figure I that there are some internal structures within drusen, which at that time was really unclear what that might be. We knew that there are lots of molecules in the drusen, from proteomics, transcriptomics, and lipidomics. Studies show that it's full of a variety of molecules. What we particularly were interested in is what is the relationship between trace elements, especially zinc at the beginning, between drusen formation and metals. And the reason for that was twofold. One is the RP choroid interface is the richest source of zinc in the body per gram of tissue. The pigmentation and the overall antioxidative processes require a huge amount of zinc to be stored in this interface. Now, of course, the drusen is not in the cells, so it's actually in the extracellular space. So what happens to zinc is a big question. And of course, again, this is a very important issue when you think about the ARAT studies, which show that zinc supplementation has a positive effect on the progression to end-stage disease. And now with the ARATS2 and new analysis, I think we have better understanding that even earlier stages like dry AMD, zinc plays a role. So what we did, we took these flat mounts to a synchrotron. And the synchrotron irradiation, what it does is non-destructively shows you elemental distribution in a tissue. So this is a flat mount. This time, actually, we used the zinc-specific fluorescent dye, which lit up every single drusen very nicely, whether it's in the macular region, the mid or far periphery. There seems to be zinc that we can label with fluorescent sensors. But fluorescent sensors can be misleading how much zinc there is. So we quantified the amount of zinc in this using the synchrotron. And what you see here is that there is very high concentration of zinc in the macula especially, but also the periphery and the far periphery as well. Now, just as a comparison, because this is difficult to understand what 380 means, this means that the concentration of zinc is likely to be in the millimolar range. It's not fully quantitative, so I'm saying likely to be. So there are very high concentration of zinc accumulation in drusen, and when you look at, as I said, both in the large areas in the Brooks membrane or in big soft drusen, smaller hard drusen, or in fact the Brooks membrane itself has significant amount of zinc accumulation. Now, zinc is very rapidly exchanged at certain compartments and very slowly dissociating in others. So what this zinc accumulation means is quite an important question. Now, what you see here is really large concentration of zinc stuck in the identifiable drusen. And because drusen is quite a permanent feature, despite some of the good progress in removing zinc, removing drusen with subthreshold laser, for example, they leave a big footprint behind. So there is a lot of zinc accumulation in the sub-RP space, and much higher than copper, although there is some copper association, or iron, but again, there is iron as well. So what is the meaning of this? Of course, we don't yet fully understand what is the meaning, but one of the questions that at that time when we studied this further was that whether there is an association between zinc or availability of zinc and the complement system. This is quite an interesting study where we didn't study complement factors in the tissue, but in isolation, so purified material. And because of this flexibility of these molecules, we were looking for solution structure analysis with Steve Perkins at UCL. The idea obviously was very interesting because complement factors were just popping up as the genetically associated component of the AMD spectrum. But it was also interesting because in previous years, in fact, probably 10 or 15 years before these studies, some people have already shown that zinc can bind to a whole raft of complement proteins. But the question is, you know, when you have a protein and you have cysteines or histidines, they can bind zinc, but is that make any difference? So what we did, we used purified C3b, complement factor H. They normally form a one-to-one complex. But what happens when we add zinc to this? So it turns out that if you add zinc to complement factor H or in fact C3b, both of them start forming very large oligomers. So obviously they will start altering their function, their mobility, and may contribute to the accumulation of complement factor H, which is now a complement system, which is now well established in the sub-RP space. The real big surprise came when we combined CFH, C3, and zinc together. Because what happened is when all these come together, they actually come out of solution. So complement factor H precipitates, and maybe that is one of the reasons where, first of all, it plays a role in the disease process, but also this is the reason why it accumulates in the sub-RP space. Further structural analysis with Steve showed that there is a fairly interesting interaction between the individual factor H molecules. Sometimes the domain 6 binds to domain 8 or domain 6 with itself, but nevertheless it's going to form a daisy chain, a very long precipitate or oligomers, which we showed in this method. So I think this is quite an interesting issue. We didn't seem to find any difference with polymorphic forms of the molecule, but in the same time, the actual role of zinc supplementation, not zinc supplementation, the presence of zinc has started to evolve in blood samples. Anneke, the Hollanders group, showed that when you add zinc, you can modify the action of the complement system. This is a bit of counterintuitive. So if zinc is accumulating and then we add zinc to the system, would we precipitate even more of this deposition? This may not be the case. Biology in the body is different than in a test tube with purified proteins. What we think happening here is then when you extract zinc into the sub-RP deposits and protein complexes, you actually generate a zinc deficiency. And because zinc is so important for the RP, the supplementation is likely to enhance all those molecular processes that are still there and should like to work and protect the RP and the choroidal cells. What we have here is a double-edged sword. In one case, it can precipitate the problem, but because the problem is associated with removing zinc from the environment, we need to supplement, and that's why we might have that positive effect in the ARET studies. Then what also happens when you do synchrotron radiation, as I showed you, you measure other elements as well. And I was concentrating on zinc, but when we started to interrogate calcium deposition, and these are the very same deposits which we measured the zinc in, what you see here, and I highlighted here with this ellipsoid, that there is 1,000 times higher concentration of calcium accumulation in these deposits than what it was in zinc. So if we talk about millimolar concentration, that's in almost the molar concentration range. Now, at this concentration, we didn't expect that calcium will be floating around or anything like that. But what we expected is probably start precipitating out with elements like phosphate. So this is just highlighting on the bottom of the slide that what could be the situation here. There is a lot of phosphate moving about inside and outside of the cells, and there is a lot of calcium as well. If they meet together under certain circumstances, they can precipitate into different form of calcium phosphate. A particular form is hydroxyapatite, and I will talk to you about hydroxyapatite a little bit more. But of course, what I mean with this figure, yes, we have a lot of calcium. It's probably in some kind of mineral form, but can be determined what that is. And as I told you before, this is when we were at the synchrotron, and if you use a different energy beam, you can do X-ray diffraction on the very same samples. And I must admit, this is the very, very first time we generated an image, and it generated a beautiful ring structure, which when we analyzed it based on the 2 theta, which is the measure of the ring structure, the blue is what we measured, and the red is the typical pattern for hydroxyapatite. And the conclusion was that at least 99% sure that it's not just a calcium, any kind of calcium crystal, but it's definitely hydroxyapatite. This, of course, has been repeated several times. And the reason why we have these ring structures, because this is a biological tissue. It's not a single crystal, what you would have in a rock or a piece of metal. So we have hydroxyapatite, and that's really weird. If you think about what hydroxyapatite is, it's making your teeth strong, making your bone strong. So why would you have a piece of bone or teeth in the back of the eye? And is this actually really relevant, or it's just some kind of fluke? Now, this is difficult to study with X-ray diffraction, because these are very time-consuming and expensive studies to run on big numbers. But that was the time when I bumped into a colleague again, Richard Thompson at University of Maryland, who is a fluorescent microscopist, and we worked together on zinc biology in the past. And as we were chatting with Richard on a grant proposal, I showed him that X-ray diffraction pattern. And I said, it doesn't make sense. I don't know what this might mean. And Richard said, oh, hold on. I actually know some fluorescent labels that can specifically label hydroxyapatite. So why don't you use that, and we see what comes back. And what came back is this picture. This is on flat mounts, where we labeled the tissue with a bone tag, a probe that doesn't exist anymore. But OsteoSense is an even better label for infrared labeling of calcification. And what you see is whether we looked at soft drusen, basal linear deposit, or small hard drusen. This is 10 micron, just to point out. We have a very, very large number of little hydroxyapatite spherules appearing in every one of them. And I don't want to overstate our findings, but really, I don't think we have ever seen a drusen without hydroxyapatite. The level of hydroxyapatite can vary between 1 and, as you can see, hundreds or even thousands. But when you look at it in a flat mount, we always do see these hydroxyapatite little spherules in any kind of drusen. This is a confocal microscopic cross-section, and what you see here is the green autofluorescence of the proteinaceous material and lipid material of a small druse, but you can see that there are multiple spherules there some that is missing, which is probably because this is not yet calcified, or this is an intermediate calcification. This may never calcify. But it's what was interesting is the inside was always empty. Of course, this could be just shadowing of the very strong shell outside. So I will come back what we did next. But what was really interesting when we looked at on flat mount is that this is the Brooks membrane, this green on both G and H images. And we can see individual hydroxyapatite spherules without the material that we call drusen stuck to the surfaces. So it seems that hydroxyapatite spherules can be there either before or without drusen. And I will come back to this in a second. To make sure that this is hydroxyapatite, fluorescent labeling is not always sufficient. So what we did, we used different methodologies like TOF or secondary ion mass spectrometry, and we could identify every single peak that is associated with hydroxyapatite that built the confidence that we are, in fact, with these fluorescent dyes, do see calcified material in these drusen. I mentioned to you that there are little empty spaces inside, at least that's how it looks like. And because the secondary ion mass spectrometry had a fairly high resolution, we were able to look at different molecular ions like calcium phosphate and others that are typical of lipids like cholesterol. And when you look at these series of little spherules here, you can see that they are all containing calcium phosphate, but they also contain cholesterol associated with them. So we think that what we have, at least in some of these drusen, they have a lipid core around which hydroxyapatite can develop, and some of them will be associated by, for example, phospholipids, where the phosphate moiety could be a binding point for the calcification. So the hard material, one of the advantages of having hard material like calcium phosphate is that you can use electron microscopy, scanning electron microscopy. And because they are so hard, sometimes they actually just split because the blade has just split them. They don't section, they split. And that's why you can identify different internal structures. And when I told you that at least some of them contains lipids, we don't yet know what other molecules can be inside. Probably we have everything hydroxyapatite, sometimes build up like a tree trunk, probably different molecules start building up on top of each other. When you look at their surfaces, that was also quite interesting, that you have different surface structures visible on scanning electron microscope. So what that might be was a question which we wanted to study, but we wanted to understand what is the distribution of these little spherules. And it was very, very stunning that after about 20,000 of these little spherules identified and measured by a student and my postdoc, Matt Pilgrim, we had a very good idea that these are not just random sizes. These are about 1.3 micron size spherules, so fairly highly determined size. We don't yet know what it means, but I feel that it does probably mean something very strongly, and maybe because they are so beautiful and round, they are linked to all those lipid molecules that are transported from the RPE to the blood or vice versa, that Christine Curcio are so eloquently presented in her oil spill hypothesis. So if you look at a cross section, here you can see the photoreceptor outer segment. Here is an RPE with the pigment particles. Even subthreshold drusen, which wouldn't be visible on any clinical machine, you can see the build of hydroxyapatite spherules. which can be a different size. Can you still hear me?
Yes,
Very well,
we can hear
thank
you
you.
well.
Okay, sorry, something happened on my screen, on the other screen. That's why I asked. So what you see here is these different surfaces and a relatively homogeneous size structure that is visible in the sub-RP space with a size of about 1.3 micron. But when you look at these surfaces, there are these little interesting hydroxyapatite spherule with some kind of material deposited on the top. And we assumed, or not, maybe not assumed, but we hypothesized that maybe this is some protein, these are some protein deposition. And in fact, if you take a small druse, which is about the size of an RPE cell, this hexagon represents about 10 micron RPE size. When you stain it for complement factor H or amyloid beta, two well-known components of drusen, what you see, they're beautifully on the surfaces of hydroxyapatite spherules. So this is quite an interesting thing. What does it mean? Why would this happen? Is actually not that complicated. But before I go there, so in the previous image, I showed you here is amyloid beta, here is complement factor H, as if it would be, one release site on an RP cell releases amyloid beta, the other a complement factor H. But it's not always like that, of course. Here is a spherule, here is amyloid beta, here is complement factor H on the very same spherule. And as you can see, some of them are overlapping, some of them are deposited in different locations. So it seems that whatever is secreted by the RPE or maybe pumped in from the blood can stick to these surfaces. So why would this be? This is actually very simple because hydroxyapatite is one of the best or most widely used chromatography medium. We use hydroxyapatite beads to separate proteins, and we believe that's exactly what's happening at the RP choroid interface, where there is a lot of exchange of proteins, lipids, and nucleic acids, and that contributes to the buildup of these molecules. So although it's not a column, but here is an eye with from Goldis Malak with age-related macular degeneration. Here in this magenta or pinkish color, you see the Brooks membrane and the fibrous material. Here are vessels in black with red blood cells in between. And you can see on the top of them where there is a huge drusen, there are hundreds of these different hydroxyapatite spherules. So there is a lot of molecule proteins, lipid can bind to the surfaces and maybe contribute to drusen formation. As I mentioned, here is an example of scanning electron microscopic image where this is the Brooks membrane. These are the pigments in the retinal pigment epithelium. These little spherules can sit there alone without any material really accumulating yet on their surfaces. So why they develop there, we don't know, but once they are there, they are potentially focal point of protein accumulation. So with a good friend who unfortunately passed away recently, Arthur Bergen, we sat down and started to discuss how we could put this together into a drusen biogenesis paper. And what we thought might be happening, that in a normal circumstances, RPE and the blood produces a lot of exchange of lipids and molecules. If hydroxyapatite gets stuck on that surface, then they start binding all sorts of different proteins, and that's how drusen are grown. And here is that picture which I showed you, a small insert from lots of drusen in a large drusen in the macular region. So what does this mean? And the real meaning behind all this calcification and drusen deposition came from a collaboration with Bailey Freund and Christine Curcio, where we had some eyes where we had very nice images at baseline and just before the patient passed away. And here I circled one of the areas that are quite typical, this little horseshoe shape. And here is in the bottom the OCT image going through three of these drusen. And you see the so-called hyperreflective pyramidal structures that we interrogated further. And the reason why we became so interested in, because of their appearance, these little dots inside, the hyperfluorescence, hyperreflection inside, and trying to understand what is the relationship. And the real striking effect is that when we took an autofluorescence image at baseline, you can see that the RPE is not happy, but it's still there. But at the final stage, all the RPE disappeared. So these structures seems to be associated with atrophy. Because this person donated the eye, Christine interrogated that both with post-mortem OCT and then sectioned them, and we found these structures, which we call now nodules, that we took further and analyzed them. So these are the multimodal imaging of that very same drusen. They are quite hyper reflective on color. They are hyper reflective on OCT, and on they are also containing some hyper reflective dots inside on infrared. And then there are other elements that on, especially on infrared, you can see lighting up like Christmas trees. So what are these materials? Because this is a postmortem tissue, we could isolate those big nodules. I showed you before about the spherules, but we also took parts of these Brooks membrane plaques and we analyzed them for elemental composition. What we found is that they all contain calcium and phosphate, and in fact, they are not 100% identical. The little spherules, apart from calcium and phosphate, also contain magnesium, while the Brooks membrane plaques are also containing magnesium, but in much less content, and their all appearance is very different. But the big nodules do not contain magnesium. When we took them further and generated X-ray diffraction on them, what we learned is that the nodules are polycrystalline hydroxyapatite, what we've seen before, but the spherules seems, because of the magnesium, are slightly different. They are calcium phosphate, but with magnesium doping, which actually is called Whitlockite, and the X-ray diffraction shows exactly that. While the low crystallinity Brooks membrane is an apatite, but it's very difficult with crystal diffraction of this disorganized structure to make more out of it. But why is this important? Because what we know is magnesium is limiting crystal growth. And maybe that's the difference between the big nodules that lead to those disruptions and the small spherules that are eventually leading to big drusen and big disruption. But at the beginning stage, they are not as damaging because of this magnesium built up inside. I was talking about the drusen in the periphery before, and this is an example to show you that, you know, here are two drusen at baseline 2 years later, and what you see here, some of those drusen, even in the periphery, goes into atrophy. So it's not just a central change, but also a peripheral change as well. Now, Wassad, I had a really nice cohort of 138 patients with these hyperreflective structures, and it turned out that all of them with this feature develop a much higher odds ratio to develop very rapid, within 1 year, progression to end stage AMD, especially GA. We didn't have enough CMV cases. So it's very, very exciting to see that calcified drusen nodules, calcified material can directly lead to rapid progression. And I think this is a very important element of it. And just before I finish, I thought that I tell you a little bit about another disease, Pseudosantoma elasticum, which is the typical calcification disease, and also about aged eyes. And what you see on both flat mounts is that there is a lot of white labeling, which is hydroxyapatite staining with fluorescent dyes. And what you see here, even in an aged eye, there is plenty of Bruch's membrane calcification, even if drusen are more on the peripheral retina. What you see here, these little dots. While of course in Pseudosantoma elasticum, the whole macular region is gradually spreading its full calcification to the periphery. So calcification is important not just for drusen, not just for nodules or spherules, but also for the Bruch's membrane calcification, which of course in Pseudosantoma elasticum leads to angioid streaks, all sorts of pigmentary changes, and the destruction of the vision by developing new vessels, complete neovascularization and scar. So this is the clinical image of this patient who after dying donated the eye. And here is a small section, here is the optic nerve. The reds represent where the blood vessels are in the choroid, and you see patches of now choroidal neovascularization. But basically, the whole of the back of the eye is completely calcified. It splits, which is the cause of neovascularization. And what's also interesting is as the disease progressing, as you can see here, it's all calcified, but it's green on the top. And that's because it's happening, the same thing happening with the Bruch's membrane as with the spherules. They start binding proteins and they will block the detection in this way, while at the more peripheral region, that binding hasn't happened yet. So it's quite an interesting thing. And when you look at the cross section of this eye, you see the pink full calcification of the Bruch's membrane, neovascularization in some cases, but most importantly, there is a lot of atrophy happening even outside of the neovascularization. So yet again, calcification seems to be linked to atrophy of the retinal layers above. I think it's important to mention that when you look at this fully calcified region, although the disease is called Pseudosantoma elasticum, saying that it's the elastic layer that calcifies, actually all layers of the Bruch's membrane are calcified. And if you go to the peripheral areas, it's much more the collagenous layer than the elastic layer in the middle that is calcified in the eye. You could ask, okay, it's Bruch's membrane, is there any drusen? If you ask clinicians, they don't see drusen. They don't probably look for it because these patients have a lot of other problems. This is the retina, the flat mount retina of that patient you saw before. And what you see is inside the retina, there is a huge number of these little hydroxyapatite spherules stuck inside the layers which are above the scars, but of course the RP is already gone, so these probably just get stuck in the retina. This is the avascular zone in the retina. And now I'm nearing to the end, so bear with me just for a few more minutes. What is interesting when you look at, and this image was generated way before these observations with the nodules started, we started to see the regular shaped little spherules, but we also saw this bigger calcium deposition with inside very strong labeling. So what it suggests to me, and I don't know whether I'm 100% right, but I think it's quite indicative, that spherules and nodules can develop in the same place. They probably remain different because they are made slightly differently, but they are reflecting what is the state of the tissue. And depending on the nutritional state of these RPE cells, if there is enough magnesium, then you will have spherules. If the magnesium disappears, you probably have a much faster or a much better growth for the bigger nodules that might be more problematic. And what we can do with this in the future, and your Christoph's group is the one who made pioneering work on this. If you look at how to detect calcification, we figured out that if you bind fluorescent sensors that change their lifetime, then we might be able to see these calcium deposits early on, because it doesn't depend on the intensity of fluorescence, but the lifetime changes. Here is two examples where a short and a long lifetime, how it differentiate. And when we did flat mount labeling, we could see two on the phasor plots. We could see two specific labels or signals. One labels drusen, the other one labels the surrounding RPE cells. So maybe fluorescent lifetime imaging ophthalmoscopy, which is now available in clinics, would be a good tool to monitor earlier changes way before these changes could lead to irreversible change. And to those who are like me, didn't study German, drusen means what you see on the left-hand side. This is this little geode with all sorts of crystalline inclusions inside. And this is how it looks in drusen in the eye. So the German ophthalmologist who termed them drusen wasn't wrong at all. Crystals developing in an inclusion, just like as it happens in geology. So I think it's a quite interesting trivia fact. So when you go to the pub, if you come to the UK, you might be able to use that in the future. And the final thing is that this attracted the attention of NIH, and currently we are running a program grant with 4 labs in the US and my team here in Belfast. So it's going to be quite interesting to see that in the next 3-4 years, how we can progress, because we now have animal models, we have cellular models that can produce calcium deposition. So a lot of things hopefully will be revealed in the near future. So I will stop here, and thank you very much for your attention. And if you have any questions, please do not hesitate to ask.
Imre, this was really wonderful, and I have so many questions that comes to my mind that I'm not sure we have enough time for them. So before I will go to other people, let me just ask maybe very quickly two simple question. If you have hydroxyapatite, obviously the ionic bonds are very, very tight, very high, low probability of solubilizing hydroxyapatite. That's why we're using 4 columns, right? So is there any chance you can think of to solubilize this hydroxyapatite? Because they're slightly different because they have lipid, most likely.
So this is a super interesting question. There is a silly answer and there is a more realistic one. I suggested to my ophthalmology colleagues to inject Coca-Cola into the back of the eye, because we know that these very high acidic conditions is the only one that actually dissolves hydroxyapatite. Now, it didn't catch on, so I think we need to think about something else. But I don't actually have an answer to that, Christoph, because I believe that even if we see the drusen disappear in subthreshold laser treatment, I don't think at the moment that the hydroxyapatite will disappear. It will still be on the surface and could be inert, but it could cause trouble as well. So I don't think there is a good solution apart from acid that would dissolve hydroxyapatite deposits.
But as you know, it has been published in multiple reports that those drusens are migrating with time. So they are not present in just one position, but with time, they move around and they locate in a different places. So if hydroxyapatite is not soluble, we can assume that it's not soluble. How is that happening?
I would like to say that they do change. That's very clear. I think we are just getting at some point to the resolution level that we might be able to resolve some of these smaller structures under the RPE. And it will be very, very interesting to see whether we can distinguish whether it is just they are still there, but the drusen, like the protein lipid component, degrades, but the hydroxyapatite is still there, but in another place it start popping up. And I think why I think this might be the case because of the mosaicism of the RPE. So the RP, we have the RP monolayer, and we think about it as a monolayer. But even in cell culture, but especially in human eye, every single RP cells seem to be on its own. They communicate and work together, but they have their own features, and they could produce very localized changes which we perceive as a moving around drusen, but it's actually just popping up and disappearing drusen.
And finally, why this is too difficult question, but now will be very simple one. Why do we have this formation of the hydroxyapatite? We have phosphate everywhere. We have millimolar, if not higher, tens of millimolar calcium everywhere, and yet it sounds like this is very specific in a specific way, even in the eye, how it is formed.
This is exactly what we are trying to target here in this PO1 grant. To give you the answer, you are absolutely right. We should be a piece of stone, all of us, especially with aging, but we are not. And I think it is, well, not saying, we know from the bone literature that there are a large number of molecules that are involved in regulating the calcification process. I think depending on where you are, whether you are in the eye, the heart, the vessels, the combination of these regulatory proteins and molecules do differ, but the endpoint is the same. So fatuins, for example, the best example or most widely used examples, they are very strong regulators of calcium deposition. And I think what happens in the RP choroid interface is not only the presence of calcium and phosphate, but the disappearance of the regulatory framework around it. And I don't have that in mind.
Thank you very much for showing this wonderful cell biology work. I'm coming from the protein misfolding background. So do you think these drusens, they are like a way of throwing up the garbage from the cell, and there is a like cell dysfunction that's causing this? I'm asking because often when the intracellularly, when proteins are misfolded, as a protective response, cells actually form larger aggregates, and it protects cell from, it is still toxic. And when you're showing the different types of drusen, is there like some drusens maybe smaller but more toxic or something like that? So I'm just curious.
This is a super good question. I really like your question, and that's one of the elements that the PO1 grant is tackling. So Francesca Marazzi is a biophysicist who is looking at, in Milwaukee, looking at the interaction between hydroxyapatite and different proteins and how does that change the protein. And you are right. If we start accumulating molecules that are more toxic, for example, to the RPE, there may be a problem. Similarly, maybe that's the good thing. So they try to collect all those molecules there that they don't want and try to encapsulate them. And that's why this good old wisdom that drusen is always associated with AMD, but it doesn't mean that drusen is AMD. So maybe that is part of the protective mechanism, trying to remove toxic molecules from the environment. And eventually, once they become too big and too complex, they might actually do harm. So we don't have the answer, but I think the interaction with hydroxyapatite of the different molecules is going to be very enlightening, how they actually contribute to the disease itself.
Yeah, thank you. I'm Dr. Kim, clinical retinal specialist and using scholar to Python. So you show the higher content of zinc and calcium in central drusen rather than peripheral drusen. But clinically, doctor, so we recommend the formulation of lutein, zeaxanthin, and zinc formulation. So should we modify the recommendation? How do you think about it?
So I'm going on that on evidence, and the evidence suggests so far that the supplementation of this complex material is beneficial. There is a complex issue here. I think we are not yet at the level of specific RP choroid interface versus the whole body, but I do think the most important element about the zinc story and the ARET study is that it seems that the patients involved in the study, they do seem to have a marginal zinc deficiency. Unfortunately, this is very typical of the Western diet these days, with all the slightly plastic enhanced diet we have, that very often we do not realize that there are deficiencies, because the deficiency is not so severe that it would be clinically immediately visible. So it is a duality. I think zinc supplementation will help your general health, and that's one of the first findings of the ARET study was that those patients on supplements live longer. But I think the original work from Newsom very beautifully showed that there is a lot of direct effect of zinc supplementation on the RPE cells themselves, their oxidative resilience and so forth. So I think there is this dual effect, and it happens that when something goes wrong, the released or exchanging zinc can do bad things as well as good. But I don't think there is evidence that zinc supplementation does bad, although there is a Canadian group who raised issues about genotype and zinc supplementation. That hasn't been backed up with other studies, but there are some ideas that maybe zinc is not as good. Maybe the 80 milligram per day could be modified to lower levels. I think ARETS-2 is now only giving 25 milligrams to address those criticisms. But as long as the evidence suggests that it's beneficial, I think the modification can wait.
Hi, Doctor. Thank you so much for your wonderful talk. I really enjoyed your images. I have a naive question, but lately has been shown that the human body is full of microplastics. So I'm wondering if there's any possibility that this drusen might contain those microplastics, or is there any way to study them? Have you thought about that possibility?
So it's a very interesting question. We did think about this because we are pursuing what might be inside these deposits. Unfortunately, so we are, the Heisenberg relationship, the smaller we go, the harder it becomes. So we can go to very small resolution, but the detection limit of these chemicals become very challenging. So we are continuing our TOF-SIMS or mass spec based secondary ion mass spectrometry work, trying to identify what are those molecules inside. It's a challenging things when you are at the micromolar level, micrometer level. But I'm hopeful that we will have an answer to you. This was the very long answer to say I'm not sure, but I wouldn't be surprised.
Hello, great presentation. So I have two questions. The first one is about, does the presence of magnesium supplement reduce drusen formation?
I don't think there is good studies so far out there to address this particular question. I think it would be very interesting to do that. It may not reduce the overall drusen load, but it might reduce whether you go into the big nodules that would produce bigger and faster effects based on the clinical data from Vasada. So I think what it highlights that there is something to do with magnesium homeostasis, and I do not know any studies that would address this directly, but it would be a very, very good idea to try to do include magnesium in these supplements and see whether they help.
Great, thank you. My second question about the differences between the soft and the hard drusen. Like, can a soft drusen become a hard drusen?
This is a question I think which is discussed in many ways. I think drusen, to me, and I'm sure a lot of people won't agree with me. Drusen is a drusen. That means that extracellular deposit. Whether this soft or hard will depend on what is the environment they are growing in. And I think Christine might want to chip in. I think her work very clearly shows that the soft drusen has a different and very extensive lipid composition, which is probably coming from the particular nature of the macula. But Christine is here. Do you want to comment, Christine?
Hi, Imre, and everyone. Thank you, Imre, for your talk. A beautiful microscopy, very convincing. So the Sarks, who did the foundational ultrastructural work on AMD in the 80s and 90s, said that soft drusen had a specific composition that was distinct from hard drusen. And our studies confirmed that. That they are very lipid rich. The best electron microscopy, which is very difficult to do, is consistent with a very different ultrastructure from what's in the hard or nodular drusen. We recently published a paper showing that nodular drusen are very distinct from the soft, from the things that we call soft. Now, what we need is a better name for so-called soft. I'm kind of aiming towards visible and invisible drusen because the soft drusen are actually part of this oil spill, the basal linear deposit. And on the other hand, it has been shown going back to the Beaver Dam study that a high load of small nodular drusen over time does increase risk for, you have to have a lot of them and it has to be a very long period of time. And the important thing about the visible versus invisible soft drusen is that it's highly focused in the central retina. It's under the fovea. So it's really linked to the biology of the fovea. And I could go on about that, but it's Imre's talk.
Thanks, Christy.
This is Dominik Lewandowski. I have three quick questions. Are there any inhibitors of hydroxyapatite formation, like chelating agents or something like that? Second question, are there any genetic factors that correlate with the severity of clinical outcomes when it comes to the drusen formation? And third question,
sorry,
how do the RPE deal with the subcellular drusen hydroxyapatite deposits? Do they like try to wrap it up, push it somewhere in one place, or do they try to digest it? Or yeah, thank you so much.
Let me start with the last one because I may forget the first two. But the most important is that the RPE is actually quite resilient as a first view. So when we did some culturing of RPE cells on hydroxyapatite beads, which you can buy because of the chromatography media, the RPE cells look okay. Microscopically, but we do now build evidence that the RP above these deposits do suffer. I haven't shared that. We hope we'll be able to publish this soon, but there are a number of effects of hydroxyapatite deposition or seeding under these cells. So I think the RPE is not happy. This is not a substance that they are welcoming, but they don't engulf it. They probably try to avoid it as far as we can say at the moment. What's interesting is again a work which actually triggered by Christine's work was that we looked at cell cultures, both pig and human primary cells, and they both can by themselves in cell culture without any other intervention produce calcium deposits. So the process is inherently there, but we don't yet know what that mechanism is, what are the exact components. This is what we are trying to tease out now. I hope this answers to some degree your question. Would you mind repeating the others?
Just the that's genetic factors and inhibitors potentially.
So the genetic factors, there may be. There is no obvious choice. We need to look at the genetic associations that we already know. So the GWAS studies has already teased out where could be the genetic factors. So I don't expect there will be different genetic factors than what we already know, but it may not mean that just because we know what are the genetic polymorphisms, that they may not have an effect on calcification itself. So I don't think there is any extra than what we know, but whether those which we know about have a contribution, that will need to be examined properly. In terms of the calcification, yes, calcification and calcium growth can be modified, and we know that from bone research. But how to implement that and what are the steps that we would need to take is going to be a big question. But that is exactly why we are excited about detecting it early, because if we could show that calcification start, let's say 20 years before, which is probably starting 20 years before serious clinical consequences maybe there is a window, maybe not 20 years, maybe 5 years, where we could slow down the progression. Another opportunity is not limiting calcium deposition, but inhibiting the binding of molecules to these surfaces, so limiting their capacity to lay down the sub-RP deposits. These are all science fictions, but these are ideas that we are trying to examine.
Hello, Dr. Lengyel. It's Rafał. I'm a postdoc in Krzysztof Palczycki lab. I'm very interested in the protein biomineral interactions. So I have a question. If you know of any potential effect of inhibition of interaction between CFH or other proteins and hydroxyapatite, how would that affect the formation, the health of the RPE? Thank you.
It's a really, really cool experiment to do. I fully agree with you. No, the short answer is no. We haven't done those experiments yet, but I think it would be very interesting to see how interaction with hydroxyapatite actually affects complement factor H activity, C3b production, and so forth. This hasn't been done yet, but I think these would be really useful addition to what we know so far.
Hi, Dr. Lengyel. I'm Maria Hołowicz from Christop. Thank you for your wonderful talk. And do you observe differences in composition with aging?
So the short answer is no, but that's not because there could not be. It's just simply we haven't looked at those. I think hydroxyapatite is the end product, so it's hard to imagine that apart from very minor modification whereby magnesium or zinc or some other molecule built into the crystal structure, that will make a big difference. But of course, as the RPE ages, what it will secrete is going to change. So building of the drusen, I think it will depend on the age. And I think it to me it was astounding to see a drusen where we have one spherule with only amyloid beta, another spherule with only complement factor H, and another one which is a combination, that it's whatever is the RP secretes at certain individuals' release sites might contribute to what the fate of drusen will be, or this deposition will be, rather than just the age. So yes, I think the age affects the RP, and therefore it's going to affect how these deposits are formed.
Hi, thank you so much for the wonderful talk. So my question is about zinc physiology in the eye. So patients that have zinc deficiency often will have symptoms related to the delayed wound healing, so acrid dermatitis, or even like neurological symptoms like anosmia, but you don't find them any ocular findings. So how do you reconcile that? Could it be similar to an anemia of chronic disease state where you have the shift of these trace minerals into other compartments?
I think this is a super interesting question. I don't have a clear answer to that. Now, I would like to add, though, that I don't think we have ever looked at whether there are ocular manifestations. So you need to remember that there is a bit of a disjoint between what I presented to you and what you can look at in clinic. And so once we have the tissue, we can start analyzing their mineral composition. And I doubt that you would try inoculate a person with anemia. So to answer that question, we would need to have the right tissue. And I think we have never examined this to the degree to answer your question clearly. So I don't think there is yet a discrepancy there, but it's an interesting question that would need to be examined further.
And we have a question from the Zoom. Stephen Sinclair. Stephen, are you ready? Okay.
Can you hear me now?
Yep.
Okay. There is now significant evidence that choriocapillaris ischemia, perhaps contributed by the short posterior ciliary arteries that arc up into the choriocapillaris, create ischemia, and that actually drusen are the byproducts of this ischemia. Have you done either culturing or examined, say, pathophysiologic specimens that can examine the hydroxyapatite or other chemical compositions of drusen dependent upon the amount of ischemia or progress of it?
Unfortunately, the short answer is no, I haven't, but we are working on a three-dimensional or microfluidic system that exactly would be able to well, exactly. Hopefully, we'll be able to answer some of these questions. It is a very, very good question. I think in my book, the contribution from the choriocapillaris and the RPE are both critical for drusen formation. So it's not RPE or choroid, it's both. And we do have some proteomics evidence where we showed that we could identify hydroxyapatite binding proteins that has never been shown in drusen because they have never been looked at, but also it's not present in the RPE, PZP placenta zone, pregnancy zone protein was one of those that when we looked at blood composition and interaction with hydroxyapatite, which is purely in the placenta and in blood, never been shown in the eye, and it's very clearly present in the sub-RP space. So the contribution from both sides, I think, is important, and I think your question is really interesting. We would need to examine this properly, how the insults that we are associating with the disease could contribute to zinc deposition, and that's the work which we are carrying out as part of this PO1 project.
Thank you very much. Excellent, and please proceed with it.
Thank you very much.
Thank you so much for that really interesting talk. So forgive me if this is a naive question. I don't know much about drusen biology, but I was kind of curious if there are age or disease-related changes in the buffering capacity of the choroid that might lead to hydroxyapatite or other mineral deposition in drusen formation. Or do you envision it kind of being like a stagnation of fluid or lipid clearance, like stagnation of fluid exchange, lipid clearance, ion exchange across membranes as well that lead to drusen deposits and mineral deposition?
This is a very interesting question. Try to answer it rapidly. One of the elements of this whole process will be, for example, pH changes. So we know from hydroxyapatite deposition that the pH will determine whether calcium phosphate remains in a more soluble form or a more precipitated form. So, it's just an example, but I think there is a lot we need to understand before I could answer this fully.
Hi, Andrew Brown from UC Irvine. Thank you so much for this great talk. I was wondering if you could help us to clarify from your flat mounts and the metallomics, the amount of deposition of iron seemed to be very low, but we have some fairly prominent people in the field who have kind of investigated the accumulation of iron in primarily the RPE and seems to be quite high. So could you help clarify that?
So I have to say that I talked about zinc and calcium because it was easier. The very simple answer is methodological. So when you are using synchrotron, the actual values are relative to the iron, not exact. So we would only be able to answer this question with certainty if we had iron standards where we knew exactly how much iron was. So I think those values are indicative that there is iron, but it's not telling you how much there is compared to zinc.
Okay. And we have last question from the Zoom audience. Vipul Parmar, are you ready?
Yes. Can you hear me?
Yep.
Yeah. So we and others have found that osteopontin is found in the sub-RP deposits in drusens, and that's an inhibitor of biomineralization. So instead of using, something acidic like Pepsi, do you have any thoughts on what osteopontin is doing? Because it's also upregulated in AMD patients. So do you have any comments or thoughts on that?
So I don't have a definite answer to that. I know about the osteopontin. In fact, we ourselves, once we saw probably your poster at Arvo, we stained it for osteopontin, and it forms this beautiful corona around the hydroxyapatite. I don't know whether this is cause or consequence, whether it just binds to it because it's able to, or it binds to it because it wants to limit further calcification. But I think this is one of the interesting questions that needs to be answered, because there are, I'm sure, internal regulatory processes that try to limit the appearance and the growth of these calcifications.
Thank you.
All right.
And second,
we had. Do you have a second question?
Yeah. Thank you, Chris. So the presence of hydroxyapatite can also induce oxidative stress, and we know that's a very preferred mechanism for pathology. So do you think these hydroxyapatite crystals are part of oxidative stress that is caused in the retina?
We don't have direct evidence, but it's a very good question and something that we are pursuing. What are the changes that are happening there? I would say that anything in that RPE choroid interface that shouldn't be there will cause a stress to the system. But that's a very general answer that I don't have an answer.
Okay,
if that
thank you.
makes sense. Thank
you.
Imre, we had a wonderful presentation by you and also great conversation and discussion. So thank you very much. Unless Professor Cooperman has the final thoughts to summarize everything. If not, Barry, are you there?
Yes, I am. That was a beautiful lecture and love the amazing images, some of the most spectacular things I've seen. And thank you for the image of a geode. Again, it's always been confusing why the German ophthalmologist, who we don't know who his name is, I'm sure somebody does, called them drusen to begin with in reference to the word geode, but that was a beautiful image clarifying that. So thank you, Imre. I learned a lot and love your lecture. Thank you so much.
Thank you very much.
All right,
Thank you
let's
very much
join.
all. Thanks, Christoph.